Okay, it's a time. So good morning, everyone. Yeah, good morning again, everyone. Welcome and thank you for joining us for today's webinar titled Design, Detect, Deliver. Yeah, Eliza from Bench to Breakthrough. My name is Azrina Ali, and I am from Epical Scientific. I am the product manager in charge for Elabscience. I'll be your host for today's session. We are proud to co-host this webinar with Elabscience, and we are very excited to have all of you with us, whether you are joining from academia, you are the clinical research or industry. Today's session is packed with expert guidance and practical strategies to help you, yeah, to take your ELISA experiments to the next level. Okay. Hi, Prof. Yeah, Prof. Chia. Okay, I'll contact you later. Okay, just a brief introduction for those who may not know us. Who is Apical Scientific? Okay, Apical Scientific is a leading life science distributor, service provider, and manufacturer. We are manufacturer also in Malaysia. We have been in the industry for over 23 years, yeah, more than 2 decades, working closely with researchers and labs across the country to provide high quality products, responsive support, and practical solutions so you can focus on the core of your research. Our collaborations with Elabscience is one we are especially proud of, and today webinar is a great example of the partnership in action. Okay, I'm also pleased to introduce our co-host for today, Miss Lizzie from Elabscience, who will be joining us throughout the session and assisting with the Q&A at the end. Lizzie, would you like to say a quick hello?
Hello,
Lizzie.
Azrina.
Okay.
Yeah, good morning,
Uh,
everyone. This is
okay.
Lizzie, yeah, from
Okay.
Elabscience Television. Yeah.
Thank you. Okay. So I'll pass to you later, Lizzie. So what can you expect today? This session will cover how to design ELISA, the selection, application in translational research, and the trend in research ELISA. Whether you are beginners or a seasoned researcher, you'll come away with valuable tips you can apply right away in the lab. Okay, so now it's my pleasure to introduce our today speaker, Dr. Sundas R. Qureshi. Hi, Doctor. Okay, the Business Development Manager at Elabscience Bio Innovation. Yeah, Dr. Qureshi has a strong background in molecular biology and translational research, and she brings deep insight into assay development and optimization. She'll be sharing a practical, research-focused approach to ELISA, designed to help you generate accurate, reproducible, and publishable results. Before we begin, just a few housekeeping reminders. This session is being recorded, and the recording will be made available approximately 2 weeks after today's webinar. I think we will send the link to all registered participants via email. If you have any questions at any point, please use the chat box. We'll have a session. We will have the Q&A session at the end, yeah, where Dr. Qureshi and Miss Lizzie will help to answer as many as your question possible. And with that, let's begin. I'll pass the floor to Miss Lizzie.
Hello, everyone. Thanks for Azrina's introduction for our webinar topic, as well as our speaker today, Dr. Sinders. And this is Lizzie from Elabscience Vision. Yeah, Elabscience is mainly focusing on the research and production of our scientific regions, like covering immunology and cell biologies. And right now, ELISA kits, we have done many services kits for our Malaysia labs and university customers. So today, Dr. Sanders will deliver more information and acknowledgement about how we can do ELISA kit successful. And she will give more technical aspects and also his thoughts about ELISA kit. So now I will hand over to Dr. Sanders because she will take more slides to share with you with the fruitful knowledge about the laser kit. And you can focus on the question that's highlights in each part because we will have the quiz part in after the presentation. So everyone gonna have the chance to win our surprising gift. Yeah. So Dr. Sanders? Yeah.
Thank you so much, Lizzie, and welcome everyone. Good morning and assalamu alaikum. Elabscience is, as Lizzie have just explained, that we are immunology and cell biology related company. So we are basically focusing on ELISA. ELISA has long been a cornerstone of bioscience, and yet its true value lies in how it connects basic discoveries to the translational outcomes. Today, we'll explore how rigorous design or a smart kit selection procedure, reliable data, and all of these things can integrate with the complementary assays and expanding the scientific insights, and how innovations in ELISA are driving the translational research. From uncovering the mechanisms in the lab to informing the preclinical studies and real world applications. By the end, you will see how ELISA delivers not just data, but pathways to discovery, validation, and breakthrough impact. Azrina has already introduced me, so you can just look at my contact information. If you have any questions during the presentation or after, so you can contact me directly on this contact information. Either email would be fine, but if you you want to contact on WhatsApp, that's also okay. All right, I just want to quickly give you a brief introduction of Elabscience. Elabscience is actually a group company. The mother company is Elabscience, which basically deals with flow cytometric antibodies. We manufacture functional assays for cell, metabolism assays, ELISA kits, and also the antibodies and proteins, which are also used as a raw material for IVD. We also have a rich platform of recombinant monoclonal and polyclonal antibody, and we are also working with nanobody by using phage display and yeast display technologies. Other than that, we also have a subsidiary brand, which is Procella by Elabscience, basically dealing with cell culture related products, including the medium and supplementary reagents. Procella is also largest cell bank in central China. And currently, we own nearly 700 primary cells and 800 cell lines. And we also have a fully equipped functional center, which deals with our daily routine work. Okay, here is just our working environment. Currently, we own about 6 different buildings covering an office area of 12,000 meters square, 15,000 meters square of the production facility, and 6,000 meters square of GMP plant. Here comes some of our research facilities. We are fully equipped with all the related research facilities for R&D, including the purification systems, cell culture labs, all the related equipments like flow cytometry, confocal microscopy, or other laboratory equipments like incubators. And we also have a well-developed packaging and filling, as well as the manufacturing plant for the cell culture media. Okay, as Lizzie has already mentioned, at the end of today's webinar, Lizzie will host a fun bonus quiz for you, and there will be 5 quick questions, and the person giving the first correct answer will win. Each of the winner gets an exclusive Elabscience backpack, so stay sharp and don't miss your chance. Winners will be announced at the end of the session, and Epical's team will contact you for the delivery of the prizes. Okay, now let's begin our today's presentation. This session will be divided into 3 sections. And we will start with looking into ELISA experiments. Then we'll look into how to select the right kits and avoid some common errors. Next, we'll explore how ELISA can be integrated with other assays for deeper insights. And finally, we'll discuss its role in translational research and latest innovations shaping the field. So in this first section, we are going to explore how to turn the scientific question into a well-structured ELISA experiment. You will learn key principles of planning the studies that are reliable, reproducible, and ready for publication. So let's start with a quick refresher on ELISA. ELISA, or enzyme-linked immunosorbent assay, measures the specific proteins in a sample using antibody-antigen interactions. It is highly sensitive and widely used in both research as well as diagnostics. In principle, ELISA captures a target protein with a specific antibody and uses a detection antibody to produce a measurable signal, allowing the precise quantification of the protein in the sample. So understanding this foundation helps us plan experiments that are accurate and meaningful. Every ELISA experiment begins with a hypothesis. Start by clearly defining what you want to measure and why. Then translate into an assay, which is design that can actually test your hypothesis. A well-planned design considers a sample type, controls, detection method, etc. This ensures that the data generated directly gives the answer to the research question and reduces any wasted efforts or ambiguous results. A strong research question is precise and measurable. Poorly framed questions are often vague, like asking what proteins are involved in inflammation, or does treatment affect the cells? These kind of questions are too broad and make it hard to design the experiments. So well-framed questions specify exactly what we are going to measure, in which sample, and under what conditions. For example, asking which cytokines are elevated in the plasma of the patient or in any sample for a particular X condition compared to the healthy controls or the treated ones. So this ultimately gives us clear direction, and framing these questions this way helps us guide the assay selection, control setup, and data analysis, ensuring the reliable and meaningful results. Not all ELISA formats are same. There are several different types, like direct ELISA, which is simple and quick and making it suitable for high abundance proteins, but it has a lower sensitivity and it's rarely used due to its limited flexibility. Indirect ELISA boosts the signal and uses a secondary antibody, which improves the detection and allows more versatility, though it requires an extra step. If we talk about the sandwich ELISA, this is a highly sensitive and specifically ideal for the accurate measurement of low abundance proteins and complex samples. So mostly in practice, sandwich ELISA is the method which is basically in practice. Now, if we talk about controls, controls are actually the backbone of a reliable ELISA result. Positive control confirms the assay is working. Negative control detects non-specific signals, and blank well helps us determine the background noise. Including the proper control allows you to interpret data confidently, identify the errors early, and avoid any false conclusions. So always plan controls before starting your experiment, not as the afterthought. Then the replicates help ensure that our results are reliable. There are two types of the replicates. One of the type is the biological replicates. These capture the variability between the subjects or the samples, reflecting real biological differences like serum or plasma. Then there are some technical replicates which show variability from the assay itself. So combining both types helps distinguish true biological effects for a particular experiment and background noise. So planning the replicates correctly also strengthens the statistical analysis and the data credibility. Proper sample handling is also critical for accurate ELISA results. Protein in serum, plasma, cell culture supernatant, or tissue homogenate can degrade if the samples undergo repeated freeze-thaw cycles or are stored improperly. So centrifugation helps remove the debris and prevents interference in the assay. Always label your samples clearly and store them at a recommended temperatures for their particular type. And gentle thawing on ice at 4 degrees Celsius preserves the protein integrity, ensuring the reliable measurements. Finally, the choosing right dilution ensures your sample signal falls within the assay dynamic range. Samples that are too concentrated can saturate the signal, while overly diluted samples may be undetectable. So what are the common strategies? Common strategies include performing a serial dilution to find the optimal range, or using the dilution ratios already recommended in the literature for a particular sample type. You can also contact Elabscience if you don't know about your sample dilution. Our technical team will help you to find the exact dilution ratio for your sample. So starting with a pilot experiment, it also helps us determine the best dilution for the accurate measurement. So sometimes we also recommend customers to do a small pre-test. And now, high quality ELISA data is not just about running the assay. It's about documenting it properly. Publication expect clear reporting of sample numbers, controls, replicates, and statistical methods. Include assay validation and any deviations from the standard protocols. Meeting these standards ensure that your results are reproducible, credible, and acceptable for the peer-reviewed journals. So to conclude this section, successful ELISA experiment begins with a strong research question and a clear assay design. Choosing the right format and including proper controls build confidence in the results. Using both biological and technical replicates allow us to separate true biological variation from the experimental noise. Proper handling of serum, plasma, and other samples protect the sample integrity. Dilution strategies include the serial dilutions and literature-based ratio helps us keep the signal within the assay's dynamic range. Finally, following the publication standards in reporting the sample size replicates and validation ensures that the data is reproducible, credible, and ready for high impact factor peer-reviewed journals. And next, we'll focus on choosing the right ELISA kits and avoid any common mistakes. You will discover how sensitivity, specificity, and proper execution can make the difference, and unclear or trustworthy results will be found. And currently, Elabscience is offering several different product lines like a traditional ELISA, Quickie Pro, Mini Sample, High Sensitivity, Uncoated ELISA, and EcoPlex. Let me explain each of them a little bit one by one. Traditional ELISA is the traditional method, and you will find many different protein targets in this product line. The sample volume is required, which is 100 microliter per well for the sandwich ELISA and 50 microliter per well for the competitive ELISA. It has 3 steps for sandwich method and 2 steps for the competitive method. And the storage can be done for almost all of the kits at 2 to 8 degrees, except for the uncoated ELISA. And if the kit is unopened, you can keep all of these kits at 2 to 8 degrees. But once opened, you can still use the kit for 1 year, but you have to keep the components according to the manual. Then if we talk about the Quickie Pro ELISA. Quickie Pro ELISA is the updated version of our traditional ELISA, because some of the customers, they were very keen to find a solution for the longer assay time. Traditional ELISA takes 3.5 hours to complete, but sometimes the labs doesn't have so much time. So we have optimized our traditional ELISA kits and came up with a solution of Quickie Pro ELISA kit, which only takes 90 minutes and just one step of the procedure. And this kit also requires just 50 microliter of volume per well. The third one is the mini sample ELISA kit. This is also based on the traditional ELISA, but this is specifically for some rare samples like mouse urine or mouse saliva, because you know that these two kinds or some other rare samples are very little to get. So it is very hard to find a large number of volume for these kind of samples. So that is why for such kind of samples, the customer can use a mini sample ELISA kit, which only needs just 25 microliter of sample per well. But it will take the same time as traditional ELISA. Then the next one is the high sensitivity ELISA kit. There are some cytokines which are not so much abundant in the sample. Their concentration is quite low. So high sensitivity ELISA kit have a very high sensitivity, and it can detect up to femtogram level of the concentration in a given sample. The fifth one is the uncoated ELISA. It is a very cost-effective method and flexible to use, because all of the other ELISA kits, they are pre-coated kits. But this ELISA kit is uncoated. So the customer has to coat these ELISA kits by themselves. And these are very cost-effective for those kind of researchers who have larger sample volumes and limited budget. So they can optimize these kits by themselves at the lab. They just have to perform the coating by themselves and incubate it for 24 hours, and they are good to go whenever they want. And just now we have also, we know that our customers are so loyal to us. So they were giving us feedback, and to support their loyalty, we have just reduced the price of uncoated ELISA kit as well. So you will be having this news very soon. because we are just about to launch it. And then the last one is the Ecoplex multiplex kit. This is a multiplexing type of kit in which we can detect several number of samples at several number of indicators at one time. You know that all the ELISA kits, they can only detect one sample at a time. So you can see if you are using the traditional ELISA, you will be taking just 3.5 hours just for one indicator or one biomarker to detect. But in case of Ecoplex kit, we are offering up to 18 different indicators to be detected at the same time. The sample volume requirement is also very little, and it just requires 50 to 45 microliter of sample, depending upon the indicators you need to detect. And the operation is also very simple. It does have multi steps like traditional ELISA, but still the operational procedure could be completed in just 3 hours. So that was all the details about the Elabscience ELISA kits. Now let's move on. Okay, now the choice of ELISA kit is not just a shopping decision, which we can take randomly. So it is actually the foundation of your results. A well-validated kits give you the accurate and reproducible data that you can trust. So in contrast, the wrong kit can introduce errors that no amount of troubleshooting will truly fix. So reliable kits add credibility to your work, making it easier to publish and more convincing to the reviewers. Since ELISA data often guides follow-up experiments and even the clinical insights, kit quality has a long-lasting impact. That is why the kit selection should be treated as a core scientific step, not just a routine purchase. ELISA kits are highly species specific, which means that they are designed to recognize the proteins from a defined species. If you use human kit or mouse samples, the antibody may not bind at all, or it may bind incorrectly and give a false positive. So this is the danger of cross-reactivity where antibodies detect unintended proteins. It is especially problematic in studies with the multiple species like xenograft or co-culture models. Although read the kit datasheet carefully to confirm that the species compatibility is fixed or not. If there is any cross reactivity or you have done the testing, the information will help you avoid the misleading results. Sensitivity tells you the lowest concentration detectable, while the dynamic range defines the window where the results are accurate. So whether your targets fall within the measurable range or not accurately indicates how close the assay results is to the true value. Kit datasheets contain an important validation data that you cannot overlook. Look first at the recovery rates. This tells you whether the kit accurately measures the target in different sample types. Linearity shows if the assay response is proportional across the concentrations, or precision include the intra and inter-assay variability, indicating how reproducible your measurements will be. If these values are incomplete or unclear, treat the kit cautiously. Always favor manufacturers that provide transparent, well-documented validation data. And understanding this information ensures that your results are credible and reliable. And there is one more thing I want to mention, that when the researchers are newly entering in the lab, they do have a kind of habit. I don't know how to explain it, but I have seen in younger researchers, maybe when I was a younger researcher, I might have done that, that we avoid reading the manuals carefully. So this is a very important step. You have to read the manual, each and every word, don't move forward until you understand everything, because it will definitely cost you your results later on. Now, there are two main types of pitfalls in ELISA, which is kit related and execution related. Kit mistakes include selecting the wrong species, ignoring the sensitivity requirements, or skipping the validation checks, simply. So execution errors occur during the experiment, such as poor pipetting technique, inconsistent washing, or incorrect incubation times. The manual will contain each and every step clearly in the Elabscience products. So just follow that, and I hope that you will not have any problem. Even the best kit cannot compensate for these mistakes. So both categories can distort the results and reduce reproducibility. Recognizing these pitfalls beforehand allows us to plan carefully and avoid wasting samples or reagents. ELISA also provide the protein label measurements, but it only tells the part of the story. Combining it with other assay gives us multi-dimensional view. That is the start of translational research. qPCR reveals gene expression patterns. Western blot confirms the size and specificity of the protein, and flow cytometry links the secreted proteins to a particular cell population. So integrating these models helps us verify the findings and adds the biological context. It also reduces the risk of misinterpretation for relying on a single technique. Using multiple assays together strengthens both the reliability and depth of your research conclusions. Now I'm going to give you a few case study examples for each of these techniques. Here in this publication is from Scientific Reports, just a recent publication, not so long ago. Here, ELISA measures the actual protein levels while qPCR detects the gene expression. Now, what happens here? Using both these techniques together allow us to connect the changes at the messenger RNA level with real protein output. For instance, in this study, cytokine gene upregulation was confirmed by increased serum cytokine levels, ensuring that the observed gene expression changes biologically meaningful. This cross-validation ensures that observed gene expression changes are biologically meaningful. It also helps identify any discrepancies where messenger RNA changes do not translate into protein changes. Combining these assays strengthens the confidence in the experimental conclusions, as well as provide a more complete understanding of the cellular responses. And this will ultimately help us to reach for the high impact factor publications. ELISA also measures the protein levels, but cannot confirm the molecular size of the detected protein. So Western blot experiments complement ELISA by verifying that signal which corresponds to the correct protein bond, ensuring the specificity. In this study example, ELISA shows the increased MUC5AC protein levels in the primary human nasal epithelial cells, which were treated with recombinant human IL-19. Western blot confirms the expression of the same target in the same cells, verifying both size and identity. By combining these methods, the researchers in this study validated both the quantity and specificity of the said protein. ELISA provides high throughput measurement, while Western blot ensures the accuracy, reducing false positive and strengthening the confidence in the results. ELISA also measures the amount of cytokines such as IL-6 secreted into the cell culture supernatant, giving us a quantitative view of the protein production. However, it does not show which cell types are producing these cytokines. Flow cytometry complements ELISA by identifying the cytokine-producing T cells, revealing the cellular sources of proteins, for example. For example, in this study here, the COVID-19 patient, they mapped a specific target, TSNE, from the flow cytometry, classifying immune cells producing a specific cytokine, while ELISA assesses overall cytokine levels before and after lyophilization. Combining these methods have linked this protein quantity to its cellular origin, providing a comprehensive understanding of immune responses and allowing us to interpret both the magnitude and source of cytokine production. It is also very important to note that at Elabscience, we are providing a wide range of flow cytometric products as well as some very good quality Western blot kits. So you can also have a look on that. So to conclude this section, what we do is we have to carefully select the ELISA by using the proper experimental execution, which is essential for the accurate and reproducible results. Integrating ELISA with complementary assays such as qPCR, Western blot, and flow cytometry, etc., enhance the data reliability by confirming the specificity and linking protein levels to the cellular sources. This multi-assay approach provides the comprehensive understanding of biological processes, which strengthens the confidence in findings and ensure results that are robust and publication ready, and most importantly, translationally relevant. Okay, finally, we'll explore how ELISA bridges the discovery to the translational applications. We will also look at emerging trends and innovations that are expanding the impact and scope of ELISA in the modern research. As we know that ELISA play a key role in translational science, bridging the laboratory discoveries to the clinical applications. Starting with the basic research, ELISA identifies the protein targets and pathways. In the preclinical model, it monitors the biomarkers and validates the therapeutic candidates. And finally, the clinical studies rely on ELISA for monitoring the diagnostic evaluation. So this pathway is actually how the translational science works and how reliable protein measurement accelerates bench to bedside translation. Using ELISA effectively at each step strengthens the overall impact of your research. ELISA is invaluable for understanding the signaling mechanisms by quantifying key pathway proteins and determining their inactivation or activation or sometimes inhibition. So, for example, in the ischemic stroke, there is some protein which is a DAMP. They activate the NF beta and MAPK pathways, which lead to some inflammasome formation and a release of IL-1 beta and IL-1/8. These cytokines act on neurons, glial cells, and sometimes immune cells, amplifying the inflammation, while caspase-1 promotes the apoptosis. ELISA can measure IL-1 beta and IL-18 levels, providing a quantitative readout through this pathway. So if you are working in such kind of study, you can also use the Elabscience metabolism assay kits, because those would also be a punching point that will give your results a credibility. In the preclinical research, ELISA is the primary tool for the biomarker discovery. It quantifies the candidate proteins in various sample types, including serum, plasma, and tissue homogenates, and even cell supernatant. So reliable detection allows the identification of molecules that correlate with disease or treatment response. These biomarkers can then guide the patient stratification in the clinical trials. ELISA sensitivity and reproducibility make it ideal for generating the preclinical data that informs the translational decisions. This is also extensively used in drug development, where it quantifies the target protein to validate the drug candidates and monitor treatment effects. In screening studies, ELISA identifies active compounds by measuring the biomarker modulation. During the pharmacodynamics or these kind of pharmaceutical studies, it tracks down the protein changes in response to the treatment. This will ultimately allow the researchers to evaluate efficacy, optimize the dosing, as well as support the regulatory submissions. ELISA provide rapid reproducible data across multiple stages of the drug development. Okay, as I just have mentioned our EcoPlex kit. So a multiplex ELISA platform like EcoPlex multiplex kit from Elabscience allows the simultaneous measurement of multi-protein or different indicators from a single sample. This will increase the throughput or conserves the precious biological material. Compared to the traditional single target assays or traditional ELISA method, multiplexing reduces the sample volume and experimental time. It is especially useful in immunology and biomarker panels where the multiple cytokines or proteins must be analyzed together. So multiplex assay, ELISA assays enhance the efficiency without compromising the data quality. The basic principle of a multiplexing assay is similar to the sandwich ELISA method. The only difference is that in case of ELISA, we use the pre-coated plates. However, in case of multiplex assays, okay. In case of multiplex assays, we use pre-coated beads or agarose beads or magnetic beads to coat the target or the antibody. So that is the only difference. Otherwise, the basic functional principle is very similar. Now, as high sensitivity ELISA techniques allow the detection of extremely low abundance proteins, it is ultra sensitive. And a single molecule ELISA can quantify proteins in femtogram range. This is critical for early biomarker discovery, low expression targets, and sometimes clinical diagnostic as well. Although at Elabscience, we are not dealing with the diagnostic ELISA kits, but we can provide you a huge portfolio of the research-based kits. Advanced detection methods such as signal amplification or fluorescence-based readouts enhance the assay sensitivity. High sensitivity ELISA extend the reach of research into previously undetectable proteins as well. ELISA data can be integrated with the omics technologies as well to build a comprehensive system biological models. Like protein quantification from the ELISA complements transcriptomics, proteomics, and metabolomic data as well. This integration depends or enables a holistic understanding of the cellular responses and the network dynamics. Using ELISA in combination with multi-omics such as transcriptome analysis, proteome analysis, or the metabolomic study allow the researchers to identify key nodes in disease pathways and predict the therapeutic interventions. It also transforms the single target analysis into system level insight. Okay, and lastly, some of the recent studies highlighted that the evolving role of ELISA in translational research. where ELISA can quantify the critical inflammatory markers such as cytokines in COVID-19 patients, revealing the immune signatures linked to the disease severity. It has also been used to detect several soluble cancer biomarkers, supporting an early detection and therapy monitoring. These examples demonstrate ELISA versatility across different disease-related sample types. When combined with complementary assays like Western blot or qPCR, it also provides both quantitative and mechanistic insights. Overall, ELISA continues to play a role in generating the data and can translate from bench to bedside. So to conclude our today's session, ELISA remains a cornerstone technique in translational research. Its ability to provide precise, quantifiable protein measurements across diverse samples bridges the laboratory discoveries and clinical applications. By integrating ELISA with the complementary assays like flow cytometry, Western blot, or qPCR, we can achieve advanced platforms and researchers can uncover mechanistic insights, identify the biomarkers, and support drug development. And ultimately, ELISA drives reproducible, impactful data that informs both science and patient care. That's all from my side. Now I'm giving the floor to Lizzie for the quiz part.
Yeah, thank you, Dr. Sinders. I hope everyone can benefit from this fruitful communications from Dr. Sinders. So right now, let's start our quiz part. So we have totally 5 quiz questions. All the questions is related to the content that Dr. Sinders just shared with everyone. And we have prepared, one gift for each winner for the quiz. So everybody, please prepare well for your finger, your mouse, your internet, everything. Okay, so let's get started. And Miss Jenny from Apical Scientific will help us to gather the winner's information in the chat box. So when you see my quiz question prompt up and you find your right answer, you can type your right answer in the chat box. So the first one will get the gift.
Yeah, and you don't have to type the complete answer. You can just type the alphabet. That's okay.
Because all the quiz questions is option.
Yeah,
questions.
it's multiple choice
A, B, C, D. So you can just type the A or B or C or D in the chat box. So it's very easy. Okay. So let's get started. First one. Quiz one is which factor is most critical in ensure reliable ELISA results? Option A, plate color. Option B is the brand of the peptide tips. Option C is room temperature alone. And option D is kit selection and validation. Okay, so can we go to the chat box? Yeah. So which one is the right answer? You can type your answer in the chat box. Yeah, very quick. Everyone is very quick. Okay, so let's choose the right answer here. Yeah, Dr. Smith. Yeah. Okay, so the right answer is D, the kit selection and the validation. And Dr. Smith, can you explain a little bit for this question?
Yeah, I have mentioned it during my presentation that the most important factor when we are starting our ELISA experiment, or even we have just to think that we might need to do the ELISA experiment. So the kit selection and it's to find out and ask your manufacturer or supplier that have you validated this kit for this particular sample? Because if the customer, the manufacturer had already done the validation, it will be very easy for you to find the right quantifiable data.
Okay, so let's check out the winner's name. Okay, because I see many answers with D, and let's check out who is the first one to give us the answer D. Up, yeah, up.
Yeah.
Okay.
Is John.
John, yeah, congrats. Yeah.
Congrats, John.
Yeah, you will receive our Elabscience logo backpack from Apical Scientific later. Yeah. Okay, so
Then
everyone, yeah, I
ready
see
for the
everyone.
next question.
Okay, I see everyone is participating in our quiz. So let's go to the second one. What is the main purpose of a positive control in ELISA kits? So I think this part is relying in the first part of Dr. Shendan's presentation. So let's choose the choice. First one, okay, is to check the background noise. And B is to dilute the samples correctly. And C is to confirm the AC is working properly. And D is to detect cross-reactive. So which one is the correct answer? Let's check out. Yeah, okay. It seems everyone got the right answer. Okay, so who is the first one? Also John? Okay, so can we give chance to other winners?
Yeah, it's better we can give the second gift to
Yong
Yi.
Yi.
Yeah.
Yeah.
Then everyone have the chance to get the gift. Yeah. The correct answer is to confirm the AC is working properly. And I think the option A is to check out the background noise. This is referred to the blank wheel, right?
Yes, exactly.
Yeah. Okay, so let's go to the quiz 3. Yeah. Which ELISA format provides the highest sensitivity and specificity for detecting a target protein? Okay, option A is direct ELISA kit, and option B is the sandwich ELISA kit. Option C is indirect ELISA kit, and the last one is the competitive ELISA kit. So which one is providing the highest sensitivity and specificity for detecting a target protein? Okay.
We already have the winner, and the correct answer is B, sandwich ELISA.
Yeah, this is also in the first part of our slides. So congrats. Yeah. Farzana, congrats.
Congratulations.
Okay. So we have 2 quiz left.
Yeah, 2 quiz.
So everyone, yeah, do your best to get our gift. Okay, quiz 4 is which statement about species specificity and cross-reactivity is correct? Okay, so please pay attention to that. Which one is correct? Okay. Option A, any kit can detect protein from any species. Option B is species specificity only matters in viral detection. Option C is cross-reactivity can lead to the false positive. Option D is cross-reactive improves sensitivity. So which statement is correct?
We have our winner, Mr. Key.
Yeah,
and
Mr. Key, congrats. Yes, option C is the correct statement.
Yeah, that if the species specificity and cross-reactivity can lead to the false positives.
Yeah.
Okay, now the
last one. Last chance to get the gift. Okay, last one. Yeah, why is this beneficial to combine ELISA with other AC like qPCR or flow cytometry, which is we have explained in the part 2 and part 3 together, right? So option A is to strengthen the confidence in results through the multi-dimensional data. Okay, and option B is to reduce the number of the sample needed. And option C is to avoid using ELISA kit entirely. And the option D is to make experiments faster but less accurate. So which one is the correct one?
Okay, we have the right answer here and our winner.
Yeah, apparently option A is the correct one.
Yeah, and the name is Zarif.
Yeah, Zarif. Congrats. So that's all for our quiz parts. So regarding the Dr. Sen's presentation and topic today, so if you have any question about how to develop your ELASA kit successful, and if you have any question about your operations and your studies, it is free to talk with us. You can send your question in the chat box, or you can tell us in the microphone. Email.
Yeah. You can just type in the chat box.
Okay, let's check out. Is there any question
during the meeting?
Yeah, during our presentation. So let's check out. I think
no, it's not for us.
Okay. Oh, for blank.
What buffer can be used? We have the first question.
Just use the reference standard and sample diluent provided in the ELASA kit is okay.
Yeah, so only the diluent that provided in the kit is added in the blank well?
Yes.
Yes. We have already set the blank well for you at the manual. You can check the manual.
Yeah.
Okay.
So. Oh, email, right?
Yeah.
The winner's email. Let's check out. Okay. So if you have any question, is free to ask Dr. Shintas right now. Also, we have our technical support female on site.
Any more questions from the floor? Oh, Lizzie, another question.
Oh.
Okay, another question.
Oh, it's not.
No, it's not. The detection principle is quite different. Multiplex ELISA has the simple principle of the sandwich ELISA method. You will get the pre-coated beads, just like pre-coated kit in the plate in the ELISA. But in case of multiplex ELISA, you are getting just the pre-coated beads. Actually, for now, there are two different types of beads in this kit, in which we are going to find out the PE fluorescence intensity rather than the OD value in ELISA. So that is the only difference. Otherwise, the operational procedure are also quite similar. And you will also, as in the ELISA, we only are going to detect just one indicator. So you will get a biotinylated antibody. But in case of multiplex, you are going to get an antibody cocktail. That is also biotinylated, but that is a biotinylated antibody cocktail, so that multiple indicators can be detected. And one more difference, which is that for the ELISA, you need to find or detect the OD value by using the multiplate reader. However, for the Ecoplex multiplex kit, you need to use the flow cytometer for detecting the PE fluorescence intensity.
Nominex. Yeah.
So
design is
we cannot provide the exact and customized fees for the customer.
Yeah, for now, yeah, that's correct. We cannot provide the customization option, but in near future, we are updating our Ecoplex multiplex kit, and the principle would remain same. but we would be able to customize the pre-coated beads as well. And the last information which I got is that we would be capable of customizing 30 indicators at the same time.
Yeah, I hope Dr. Sen answered your question. Yeah, currently for the customizing, we can customize the panel for you, not the beads.
Yeah.
Okay, any other questions from our participants?
Any more question from the floor? I think that's all.
Thank you.
Thank
you,
you
everyone.
very much. Okay, thank you, thank you. Wow, what a fantastic presentation from Dr. Sundas. Thank you very much.